I'm gonna talk about transcription in this video. Before that, I'll do a little review. We all know about central dogma given by Francis and Crick, which says the information flows from nucleic acids to proteins, and mostly the cases DNA goes to RNA via transcription and RNA goes to protein via translation. These two words, transcription and translation, is kind of easy to mix up. So we can remember it like this. In everyday vocabulary, we use the word translation in the context of languages, right? So even here, the information is flowing from the language of nucleotides to language of proteins. So that's translation. And transcription is essentially rewriting the message which is in DNA in the form of RNA. And in this video, we are going to deal with transcription. We know that the transcription is the process of making RNA from DNA. But unlike replication, both these strands are not copied. Only single strand of DNA is transcribed to RNA. Why is that? Let's see what happens if both these strands are transcribed. So I'm going to take this portion of DNA. I've written it down over here. Just an example. So from the 5' to 3' end, we have AAA, GGG as its base, nucleotide bases. So let's see what happens if that strand is being transcribed. So complementary to A will be U, uracil, because it's RNA, U, and complementary to G will be cytosine. So now let's take the 3' to 5' strand of DNA, that is thymine, cytosine, and make mRNA from it. So thymine is going to bond with adenine. Please pardon me, that's A. And cytosine is going to bond with guanine. So guanine. So what are we seeing here? One segment of DNA is giving rise to two different mRNA molecules, which will essentially make two different proteins. So it complicates the whole genetic machinery, and that's the reason why mRNA should be single-stranded. There's another reason which I won't go in detail, but if mRNA is double-stranded like this, the translation process becomes difficult because over here mRNA is not open, and in translation it is required for RNA to be open, so that there's this other molecule that can come bind to this RNA on one end with nucleotide base pairs complementary to mRNA, on the other end with amino acids residues, and make proteins. So if it's double stranded like this, it's not going to be able to bind to the mRNA, and it's difficult. So that's the reason mRNA is single-stranded. So the next question is which strand to copy. To answer that question, let's first talk about what we need for transcription to happen. Obviously, we need DNA. As you can see, there are three portions of DNA over here. One is called the promoter, the other one is called the structural gene, the other one is called the terminator. All these three put together is called a transcription unit. So let's talk about each one separately. So promoter is nothing but a bunch of nucleotide base pairs located on the upstream of the structural gene. That is few base pairs before the actual gene to be transcribed starts. This is kind of an important part because this is where the enzyme that's catalyzing the process of transcription binds, and the promoter is the one that kind of gives the green signal to the enzyme, says, okay enzyme, you can start the process of transcription. So it's kind of important. And this is the gene that's actually going to be made to mRNA. So yeah, if this is your mRNA, this is because of the structural gene portion. Okay, and termination is terminator. I think it's a sequence that's going to help in stopping the process of transcription. Next that we need is the enzyme, right? The yellow color one that's binding to the promoter. The enzyme used in the process of transcription is RNA polymerase. Just like its cousin DNA polymerase, it also follows the rule of complementarity and also transcribes only in the 5' to 3' direction. I'm just going to write RNA poly. I hope you understand. So it transcribes only in the one direction. So now it kind of answers our question which strand to copy, right? If the enzyme goes in 5' to 3', the mRNA is ought to be made in that direction, and the strand complementary to it is 3' to 5'. That's going to be used as our template strand. So the strand that's used to make the mRNA is called the template strand. So that's our 3' to 5'. So I've just written some example over here. So this is our template strand. If we make mRNA, the bases are going to be like this. Yeah, I'm going to draw over here. It's going to be A because you have T over here, over there. And instead of CC is going to bond with GG. So this is similar to this, the 5' to 3' strand, and thus this strand is called the coding strand, and all the references are made with respect to the coding strand. For example, the promoter is located on the 5' end of the coding strand. Now over here I would like to talk about the differences between the eukaryotes and prokaryotes of the mRNA that's being made. See, in eukaryotes, the mRNA made will code for only one polypeptide. But in prokaryotes, the mRNA being made can code for more than one polypeptide. See, if this is the mRNA, this is for eukaryotes. But for prokaryotes, it's going to be like this. This is one portion of mRNA coding for one polypeptide. This is going to be coding for another polypeptide. So this is prokaryotes. So if it's only one, it's called monocistronic. Cistronic means a portion of gene coding for a polypeptide. And if it's many, then it's called polycistronic. It's poly. I kind of digress from the topic, but this is kind of important. So the actual process of transcription takes place in 3 steps. Initiation, meaning to start, elongation, to make the mRNA long, and termination, that is to end. So let's begin with initiation. So the first step in the process of transcription is initiation. Over here, the enzyme RNA polymerase, which I am depicting in yellow, is binding to the promoter and initiates the process of transcription. This enzyme scans its ways through the DNA, unwinding the DNA, and finds the promoter region. I want to add in little details over here. So I've zoomed in the portion of promoter region over here. As you can notice, I've written some numbers like -10 and -35. I want you to think of it like a number line. So I'll draw a number line. Assume 0 is the first nucleotide base pair that's gonna get transcribed. 10 units to the left of it will be -10, and 35 units left of it is -35, right? Okay, I'm extending the number line. So this is going to be -35. So essentially these are nothing but the distances from the first base pair that's transcribed. So why is this important? Because apparently -10 is the region where the RNA polymerase is going to bind. and initiate the process of transcription. I'll come to minus 35 in a while. So unlike DNA polymerase, RNA polymerase does not take any help from other enzymes to unwind. If you remember, in replication, helicase does the unwinding part, and the DNA polymerase just added the nucleotides. So don't you think even RNA polymerase needs some help? Indeed, yes. It also associates temporarily with a factor called sigma, which binds to the minus 35 region of the promoter, and these two together form initiation complex because it's important in the process of initiation, and successfully starts the transcription process. And after initiation, it dissociates from the complex. And you can over here see there are two steps to initiation. The first one is the closed promoter complex. As you can see, the sigma factor and the enzyme form a complex. I have deliberately drawn it big to show that there's no gap between them, even though they are in the minus 10th and minus 31st position respectively. Then the next one is the open promoter complex, formed when the enzyme RNA polymerase moves its way through the DNA and unwinds the DNA and forms structure called transcription bubble because it's shaped like one, and it's called open promoter complex because it has opened the DNA structure over here. So now you can ask me, how do we know that it's binding to minus 35th and minus 10th? If you're really interested, you can go look up for DNA footprinting, which is the experiment done to identify the regions of DNA bound to proteins. So sorry about the spelling. Yeah, that's about initiation. Elongation is just lengthening of mRNA by addition of nucleotides by RNA polymerase as it moves through the DNA, unwinding it. The energy for this process is obtained from the nucleotide itself. As you can see over here, okay, I'll use some other color. The nucleotide base releases phosphate as it's added to the chain. So this phosphate is used for energy purposes. I would like to talk about the speed of rate of the enzyme over here. The rate of RNA polymerase is around 60 base pairs per second, which is okay. But when compared to DNA polymerase of replication, which is like 1000 base pairs per second, it's a snail. Can you think of a reason why? The reason is actually very interesting. RNA polymerase has two major problems. The first one being doing everything on its own. It's unwinding the DNA, it's adding the nucleotide base pairs, which creates strain on the DNA and makes the process slower. The other one is it has this huge baggage with it. So every time it moves forward, it adds a new base pair to it and becomes heavier, and thus it becomes slower. So this is the process of elongation. Coming to termination. The RNA polymerase moves through the DNA like a train moving in a like this, until it hits brakes. So what's a brake over here? It's a bunch of termination sequence, over here, for example, that helps RNA polymerase to stop transcribing. But interestingly, the RNA polymerase moves past it, transcribing those sequences as well in the mRNA, and looks something like this. The most interesting part is the base pairs that are transcribed are palindromic sequence. Palindromic sequences are those that read the same when read from left to right and right to left, like something like Malayalam. For example, take palindromic sequence GGCC and over here, when read from 3' to 5', it reads GGCC, and the same thing over here, and they form hydrogen bond, and over here like this, and makes it difficult for RNA polymerase to move forward. The other part is, over here I'm erasing this, hairpin bend structure is going to form complex with a protein called rho protein. Remember, during initiation there was a protein called sigma factor, which helped the RNA polymerase to start the process. Similarly, to end the process, it's taking help from this protein called rho protein. So it looks somewhat like this, and this complex is kind of very heavy, and RNA polymerase cannot move forward with this, and so it stops the process and mRNA falls off. So if you're a prokaryote, that's all. This is the end of the process. This is your mRNA, and this can go for protein synthesis. But we are eukaryotes and we have some additional processes to be made. Over here, this is our mRNA, and it's not called mRNA in eukaryotes, it's called pre-mRNA or precursor mRNA. Makes sense because we're going to make some modifications to it. The other name with which it is called is hnRNA, which is heterogeneous nuclear RNA, which is going to undergo processing to be made into a mature RNA. And that's what we're going to see next. So, in the eukaryotes, the precursor mRNA that's being synthesized undergoes modification, and it's called post-transcriptional modification. The first change that happens to an mRNA is something gets added in the 5' end or at the head of the mRNA, and that is a modified guanine. It is modified because it has a 7-methyl group to it. So a 7-methyl guanine gets added at the head of the mRNA, and since it's getting added at the 5' end, it's called capping. And remember, this is the first step that happens in a post-transcriptional modification. And the next thing that happens is something gets added at the 3' end. That is a bunch of adenine molecules, around 200 to 300 nucleotides, and since it's added at the end, it's called tailing. And the adenine molecules is called a poly A tail because there are many adenine molecules being added. So why are these modifications being done? These are done to protect the mRNA from degradation via enzymes like exonucleases, and also it helps in movement, transport of mRNA from nucleus to cytosol for translation to happen. Also helps in the process of translation by helping it bind with the ribosome. So the next modification that we're going to see is splicing. Remember when we talked about cistron, I told you eukaryotes are monocistronic. That is, the one segment of mRNA, say this green one, codes only for one protein or one polypeptide, as opposed to prokaryotes, which are polycistronic. But the problem with this eukaryotic monocistronic gene is it contains some interrupting sequences that does not code for anything. For example, if this is your mRNA, the white part is the region which is coding for a protein, and the red ones are the ones which are not coding for any polypeptide. So it's like this. So we don't want the red portion to be translated because it's not coding for anything. So what do? We remove the red portion, right? We remove the red portion and put these white portions together to form a mRNA. These red portions, which are the interrupting sequences that's not coding for anything, are called introns. There's no gap over here. It's just me trying to write with the new tool pen. The interrupting sequences are called introns, and the ones that are actually coding for a polypeptide are called exons. They're called exons. So these introns are removed from the mRNA, and exons are put together in an order and they are sent out. The mRNA, processed mRNA, is sent out of the nucleus to cytosol for translation. So this is the processed mRNA. So as you can see in this picture, the cross markings are the introns over here, which are removed. They are like snippeted out with the process of splicing over here, and they are moved to cytosol for making proteins. So that's about post-transcriptional modifications. So here's just a picture about the summary of the processes that just happened. So inside the nucleus there's transcription, and the mRNA after being modified is transported out of the nucleus for translation to occur. So these are some questions which are relevant to this topic and are being asked in competitive exams or NCRT exams, CBSE exams. I'm sure you can find answers to all these questions in this video. Try solving them and hope you had fun watching the video. Thank you.